Good day, everyone. On behalf of my fellow researchers, I will be presenting our study in silico analysis of various fungal, secondary metabolites, and antiretroviral drugs and its molecular binding to virus proteins involved in cellular attachment, fusion, and replication. The Nipah virus, or NIP, is considered as a highly pathogenic zoonotic virus that belongs to the family Paramyxoviridae. NIP has a broad species tropism with the fruit-eating bats as its natural reservoir. Pigs, horses, and other domesticated animals serve as the intermediate host for the virus, exhibiting mild severe disease. At present, there are no NIV-specific vaccines approved for use in humans, and management of NIV outbreaks is mostly preventative. The genome of NIV contains a negative sense, single non-segmental RNA, which consists of 6 transcription units that encode for the viral structural proteins and non-structural proteins. Among these structural proteins, the glycoprotein G mediates the viral entry in the F-ring receptor of the host cell, and the fusion protein is necessary for cell fusion. The F and G protein complex assists in the viral entry of the virus. Meanwhile, the phosphoprotein is essential in the viral replication of the virus. As a contribution to the global pursuit of finding bioactive compounds with anti-NIV activity, these studies focus on exploiting fungal-derived secondary metabolites and antiretroviral drugs as potential drug prototypes in silico. So the methodology is divided into 5 steps. First is the protein target preparation. We selected 3 viral proteins of the virus, the glycoprotein, the fusion protein, and the phosphoprotein. These 3 viral proteins are involved in cellular attachment, fusion, and replication of the virus into the host cell. Their amino acid sequences were extracted from the NCBI database, and generation of the protein structures were done using the Swiss model software. The second step is the ligand selection and preparation, in which we selected 49 fungal-derived secondary metabolites, as well as 14 antiretroviral drugs, for a total of 63 screened ligands. Canonical smiles of these ligands were obtained from the PubChem database. Minimization and lacking preparation is performed in the UCSF Chimera platform through the addition of the missing hydrogen atoms and appropriate charges to the structure, employing the gas-hydro charge method computed using the Amber's antechamber module. The procedure for molecular docking, the procedure for molecular docking is performed in the UCSF Chimera platform through the addition of the missing hydrogen bonds and using flexible ligands into flexible active site protocol, in which ligands are allowed to be flexible and torsion within a grid box, enclosing the ligand binding cavity of each enzyme, which are predicted using the residue depth algorithms. The binding energy threshold of the top ligand scorers were selected based on a negative 8.0 kilocalories per mole minimum binding energy score, with the presence of favorable binding interactions. Biopia Discovery Studios elucidates the molecular interaction of the virus-protein ligand complexes and the 3-dimensional structure of the complex. Of the 63 ligands docked against the target proteins of NIV, 3 fungal-derived secondary metabolites yielded the highest binding energy: alkaloids norphenadylin A and phenadylin B, and the polypeptide isohydrochromine B1 showed the highest binding affinity on the glycoprotein G, fusion protein F, and phosphoprotein P of NIV. Interestingly, phenadylin B, a fumiphenazoline alkaloid, exhibited multi-target characteristics, both having significant binding energy against the fusion protein and the phosphoprotein as seen in the table. We have here the result of the postdoc analysis. There is a 3-dimensional surface representation of phenadylin B docked to fusion protein. Pi stacking, despite being the only bond present, is sufficient to provide stability and high binding affinity to the F protein due to its non-covalent attractive nature. A parallel space type of interaction highly stabilizes the protein-ligand complex. On the other hand, phenadylin B yields the highest binding energy
You have
against the
1 minute.
high binding scores due to the number of amino acids it interacts with. In conclusion, fungal-derived secondary metabolites yielded the highest binding energy scores on the glycoprotein, fusion protein, and phosphoprotein of NIV involved in cellular attachment, fusion, and replication. Phenadylin B showed multi-target characteristics due to its favorable binding scores with the proteins F and P, and strong favorable binding interactions are predominantly charged and hydrophobic interactions, conferring stable protein-ligand complexes. And it is recommended to explore the application of the top-rank regions about their antiviral activity against NIV in vitro. So thank you for listening, and that's all for my presentation.